Good evening, or good afternoon. All right, thank you for coming to the second Climate Cafe. We've got about 100 people online joining us for tonight, so this is really great. I'm Deanna Udell. I'm the Director of Communications and Public Relations here at the Rosenstiel School, and we're glad you could join us, and we're not going to delay because I think there's going to be a lot of questions. This is relatively very new research that we're very excited and very interdisciplinary. So I'm going to go ahead and pass it over to our new dean, Ben Kurtman, who many of you met last week or 2 weeks ago, and he's going to say a few remarks.
Thank you. So despite Deanna's efforts, my job is to delay. That's what deans do. That's one of our core competencies. Thank you, Deanna. Really heartfelt thank you for coming, both online and in person. It's really great to see all of you here. One of the things when I was thinking about taking this position as dean is these events themselves. I really like making sure I carve out time to attend and making sure I carve out time to welcome you. This is one of the fun things that a dean gets to do. I have to confess there's a lot of unfun things the dean has to do, but this is one of the fun things. Tonight I'm really, I was going to make a joke, but it would get me in such deep trouble with Martin. I'm not going to do it. Right? I'm just not going to do it. But tonight's talk is Hidden Engineers of the Deep: How Fish Shape the Ocean's Carbon Cycle. And I really want to underscore how important the ocean's carbon cycle is. As we try to understand, I'm a climate modeler, and so I want to think about how to model the future of the way the climate system works. And one of the big challenges we have is understanding where the carbon goes. And a big part of that problem is the ocean. And so everyone just assumes all the ocean does is take up carbon infinitely, and it's not going to do that. And in fact, there are parts of the ocean, and we don't fully understand it, that the carbon burps. The ocean burps that carbon back out. And so the research that we're going to be talking about today, or that Martin, Amanda, and Rachel are going to be talking with Jenny about today, is how fish are such a critical component of that carbon cycle. It's really important, and it's the underpinning of what I like to do in my research. So they're helping me, even though I'm in a totally different department and they never talk to me. So in my usual delay tactic, there's a couple of things I do want to point out. This is just a fantastic example of the power of interdisciplinary science. Amanda, who's going to be up here, started out in a very different area. She has completely transformed her research portfolio, and a big part of that transformation is the interaction with biologists. So here's a marine geoscientist working with biologists. As a scientist, when I think about that, being able to do interdisciplinary science is very hard, very challenging. You have to learn how to speak a common language. We don't speak common languages. I wouldn't, if I talk to Amanda about science, she has no idea what I'm talking about, and I have no idea what she's talking about. So it's really hard to collaborate. And so that being successful in interdisciplinary science, that's something we want to be the world's leading experts on here at Rosenstiel. But this group of scientists are leading the way. They're defining how that needs to be done. And there's a second part of that I want to emphasize, is that there are some really great students in this group that are participating in that interdisciplinary science. So these students are getting hands-on experience in doing interdisciplinary work. That sets them up for whatever else they're going to do. If they're going to go on and be a lawyer like Bill Galway, unfortunate for them, if that happens, they know how to work with other people to be successful, how to find a common language, how to find common goals. So the work that's being done here has, not just in terms of the science, but in terms of training people to have the right skills to be successful out in the world, no matter what they do. This is really groundbreaking stuff. So I want to congratulate them. I want to take a moment to thank some of our team members. I want to call out Katie, Sarah, Alexandra, Hannah, McKenna, and Jenna. These are some of the unsung heroes that make sure these kind of events happen. So thank you, folks. You're in there somewhere. I wanted to thank you. I also want to thank our sponsors, Mr. William Galway, or Bill. Bill has a knack for finding where there's a need here at the Rosenstiel School and figuring out how to help us solve that need. And so, Bill, we're so grateful for your support throughout the years, this event, and everything else you do. Thank you, Bill. It's like when I get into trouble, my first thought is, oh, I better call Bill. So the other people I want to thank is the Self-Help Credit Union and Sam Chester. They're again here today. Thank you, guys. Please, yes, raise your hands. You know, and their support of this event goes deeper. I overheard a conversation, oh, I'm about to give you the notice of our internships that are available. So these guys are they're really trying to help us, not just in this dissemination of all the great work that we do to the public, but, you know, helping our students through internships, and that's, you know, just really, really appreciated. I have a couple of other hats here at the university. Boy, I am wasting people's time. I have a couple of other hats here at the university, and one is through the Cooperative Institute for Marine and Atmospheric Studies. We call it CMAS. It's a really important mechanism here at Rosenstiel. It's how we reach out and collaborate with the federal government laboratories across the street. It's a $310 million project. It's a huge project. We're really excited about that. And the second role I happen to play is the director of that institute. Someday I'll give up some of these hats. But fortunately, the director and the dean are in collusion. And so the director and the dean have decided that the director should also support the Climate Cafe. So I want to thank the director of the Cooperative Institute. Oh, that was a long story. So finally, I want to thank Jenny Staletovich. You know, if you ever get a chance to chat with Jenny, she's so knowledgeable. She's such a joy to talk to. I always walk away sort of giggling about the conversation and feeling much smarter. So please find an opportunity to chat with Jenny. She's just a gem, and I want to thank those folks at WLRN, our friends there, for allowing us to have Jenny for the afternoon. So I will read this verbatim because Deanna wanted me to read this verbatim. I know, I'm a pain. I hope tonight leaves you informed, inspired, and wondering how you can get involved. I do hope that's true. Whether that means supporting research or simply spreading the word about the work being done right here at the Rosenstiel School. Join us at future events for that. And we really mean that. You can tell this is fun for me. I'm having a great time. And I haven't even seen the science yet, and I'm still having a great time. So please come and enjoy us. Enjoy being here, take part, ask lots of questions. We really do appreciate that. Bring your friends and family. Please join us in our mission to create knowledge. I know it sounds kind of ivory tower academic, but it's true. Our job is to create knowledge, and importantly, we have to disseminate that knowledge, and we have to package and disseminate that knowledge for the public good. And this is one of those events where we're taking the knowledge we've created, and we're packaging it so it's actionable for you. It affects your lives, your thinking, how you vote, how you do things. And that's really hard to do, and this is what we're trying to do today. So thank you for all coming. I will no longer waste your time.
We're way offshore from Miami in about 1,100 feet. So now we have, look at the bottom here. You see that, all that contour there, that's the money. We are now positioned where we want to fish. We're just waiting for the temperature in our holding cooler to drop down. We bring them up fast as we can. And the trick for survival there is that we keep them at their habitat temperature. Target species in the boat. Circle hook right in the corner, as it's supposed to.
So this is our setup for the Rosies. We've got 3.
calcium and magnesium is ingested with the seawater they drink. They have to drink seawater. There's no other source for hydration when you're a marine fish. So the CO2 that the animal is generating through its metabolic processes is converted to bicarbonate, and then that bicarbonate is transported from the blood into the intestine. And in the intestine, it then shifts, because it's in very high concentrations and pH is high, it shifts from bicarbonate to carbonate. Now that carbonate then combines with the calcium and magnesium to form this magnesium-rich calcium carbonate. And so what you're seeing here, this is what the fuss is all about. So that carbonate there is what's produced in the intestine by these animals and then released into the environment at very large quantities at a global scale. Could be the biggest contributor to inorganic biologically produced carbonate in the ocean.
Come on up, Jenny, Martin, Amanda, and Rachel. We'll just put up the QR code if you want to get more information on their bios. Just scan that information and it will take you right there. Thank you.
And can everybody hear me okay? Okay, excellent. Thank you. Welcome back, everybody. I will not waste much of your time either. I do just want to say a few words, which is, as a reporter, when you hear about new science or a new story like this that has big impacts, we get really excited. And so when I first heard about this, I think Martin and Deanna contacted me, I was like, ichthy what? And then once they explained it to me, it was just really exciting. So I'm hoping that you'll learn as much as I did from these guys. So Martin's gonna start us off by giving us the big picture.
Thanks so much, Jenny. Yeah, this is on. And Ben, thank you for the very kind introduction leading up to this. I don't think you wasted anyone's time, and that was just right. Some of you may have seen in the reception area a couple of tanks. Those tanks contain toadfish, and that's the animal in the back here. That's not really what we're going to talk about mostly today, but I'm showing the toadfish because that's what we work on most of the time. And the photo here kind of shows what the fuss is all about. That's an individual toadfish. And the white specks next to that animal is the calcium carbonate that the animal has produced. Now, this photo is staged. This animal did not produce all this carbonate. The carbonate here is the product of 3 fish over 2 days, just to be completely transparent and honest about the magnitude. But as you can imagine, on a global scale, this can actually amount to a lot of calcium carbonate produced in our oceans. And that's what we're going to be talking about today. So the question, of course, is, well, one of the questions, and the first question we attacked is, why are these fish producing these carbonates in the intestine? And the first clue to that can be seen in the x-rays here. These x-rays show a flounder in freshwater and seawater. And that's one of the cool things about flounder. At least some of the flounders, they can tolerate both freshwater and seawater. The top x-ray is that one particular fish in freshwater. And if you're looking at the viscera, this is the area where the intestine and the stomach is, you can see that it is completely transparent on this x-ray. There's nothing there. That's another advantage of the flounder from the perspective of this study. 3 hours later, after we put that fish in salt water and let it sit in salt water or seawater for 3 hours, we x-rayed the same animal again. That's the bottom x-ray you're looking at. And what you can see now in that area of the viscera is these x-ray opaque structures. And that is the calcium carbonate that's formed in the intestine over just that 3-hour period. So this has to do with living in salt water. And I mentioned this briefly in the video you just saw, but when you're in salt water, when you're a marine fish, you dehydrate continuously because of the high salt concentration in seawater. The same is true for us. If you're swimming or on your beach, you're dehydrating, not just because of the sun, but also because of the high salt concentration in water. What marine fish are forced to do to compensate for that is drink, and they only have access to seawater, so they're drinking seawater to rehydrate. That's not something I would recommend for any of you. We are incapable of doing that. In fact, it would dehydrate us further. Marine fish are among the only vertebrates that can pull this off. And I've spent, it's perhaps a little sad to say, but I've spent about 30 years studying how they're able to drink seawater and actually hydrate from that source of water. And one of the tricks they do is they take up salt from that water first. They take that up in the esophagus and the anterior part of the intestine. And that salt is then eliminated mainly by the gills. That reduces the salt concentration of the fluids in the intestine and allows them to take up water. But one of the problems with that water is it contains a lot of magnesium especially, and also calcium. And to eliminate that problem, the fish transport bicarbonate from the blood into the intestine. That bicarbonate combines with the magnesium and calcium and eliminates that from solution. And that further assists water absorption. And without this mechanism, marine fish would not be able to regulate their water balance in seawater. So this is essential for survival of about 13,000 species of marine fish. Without this process, there would be no marine fish as we know them today. So this is super important for the survival of the largest group of vertebrates on Earth. It's also important for the carbon cycle, as we'll be talking about shortly. So in 2009, we took a crack at estimating how much carbonate are produced by marine fish globally. And we did this based on 2 estimates of global fish biomass. And as you can probably imagine, estimating how much biomass of fish is present in global oceans is not trivial. And of course, it's associated with some uncertainty. But we got a number for that. And then we had numbers on the production rate of this ichthyocarbonate, so the amount produced per day per fish, and we multiplied those 2 numbers. And at that time, that data suggested that about 15% of the global biogenic or biologically produced carbonate in the oceans is coming from fish. So that was a significant number. We were excited about that. However, since then, there has been revisions to estimates of the global fish biomass. And you're seeing that in the bottom diagram on the left. The bar in the red is that original study from 2009, and the remaining bars are subsequent studies, all summarized in the paper we published recently. And as you can see, most of these estimates are significantly higher than our original estimate, which of course means that the original estimate of carbonate production by fish is too low. What you can also see here is that for many of these studies, the claim is that the majority of the biomass is from fish in the mesopelagic zone. So that's fish that are residing between 200 and 1,000 meters. And if you're looking at the diagrams on the right, this is not from our work, but this is from work in the peer-reviewed literature. Those are sonars, and they are showing the density of fish at depth. And if you're looking at the top, that's the North Atlantic, just as an example, you're looking at the distribution of these fish biomasses over a 24-hour period. And what you'll see is that at night, that's the first quarter and the last quarter on this figure, a lot of this biomass is shallow, whereas during the day, a lot of this biomass, or more of this biomass, is deep. And that is the mesopelagic fish we're talking about. They do these vertical migrations and probably account for more than 90% of the global biomass of fish. So when it comes to understanding how much carbonate are produced by these fish, we realize that we need to study animals from that environment, ideally mesopelagic fish. That, unfortunately, is a challenge because most of these mesopelagic fish are small. They're caught traditionally by trawls that are trawled for hours at depth. And as a consequence, when the fish come up, it looks a little bit like that top image there. It's all smushed. Some work has been done on fish surviving that process. And a lot of credit should be given to the workers that have done that work. However, it is done on animals that are severely impacted and probably dying. So that data should be interpreted with some caution. So we went looking for an alternative to these smaller mesopelagic fish. And I'm not going to go into the details now on how we came across this animal, but we can talk about that later. But the bottom left image there is a black-belly rosefish. And black-belly rosefish live at that depth. We catch them at about 1,300 feet, as you saw in the video. And so they're in the environment where these mesopelagic fish are. And we therefore assume that they're going to behave very, very similar to these mesopelagic fish. They are subjected to the same pressure, the same temperatures, the same general environment. And in fact, when we dissect these fish, we often find mesopelagic species as prey in their stomach and intestine. So we were able to, as you saw in the video, catch them and get them to the lab and do measurements on their production rates and the content of this carbonate in the intestine. And the data you're looking at on the right-hand side here, the first panel is showing the presence of these carbonates, both in animals that were dissected immediately after capture and in animals that were kept in the lab for weeks. And as you can see, they contain carbonate in the intestinal fluids. And in fact, the amount they contain in the intestine is very, very similar to what we see in animals collected at shallower waters. So this is good. It suggests that these mesopelagic fish behave the way all the other species do, and that our assumption that their biomass is contributing globally the way we assumed is justified. The small panel on the right-hand side is showing the excretion rate. So this is the amount of carbonate that's passed from the fish to the environment, say per kilo of fish per day. And the red dot there is the prediction of the rate we would expect based on the work we did in 2009. So again, this black-belly rosefish that we believe behave like mesopelagic fish produce ichthyocarbonate and excrete it at a rate that's very, very similar to what we'd expect from other species. And that obviously has immense implications for global carbon cycling. We're going to talk more about the consequences for the carbon cycle when we get to my colleagues here.
So, Martin published a paper in July that's sort of where you reach these conclusions, and it was, I think you're underselling it. It was sort of a major finding to confirm that this kind of carbon cycling that was happening with fish in shallow water was also happening at the mesopelagic level, where there's much more fish, right? So if you could, let's put this in context of how important this is in terms of the carbon cycling. When you have the biomass, the fish biomass in the oceans, you take it away. It's like huge in this layer. So that is why this isn't just another sort of step in understanding. It could be a major step in understanding in how we model climate change.
Yeah, so I'm not going to steal the thunder from my colleagues here that are going to be talking about some of this detail.
Okay, sorry.
But as I mentioned, the 95%, the jury is still out on whether it's 90 or 95% of the global fish biomass is at depth. It is the mesopelagic zone. So all the work we do on, say, toadfish and other shallow water species is super interesting, and it's taught us a lot. But when it comes to estimating global production and the question you're after, how important is this to the carbon cycle, we really need measurements on the mesopelagics. And we haven't given up on that, but since that's very difficult, we started with these black-belly rosefish that offer a unique opportunity to study a process that has not been studied before.
Well, that you mentioned that you've studied this for a long time, and when we talked, I thought it was relatively new, and you said, no, we've known about it since like the 1930s, but there seemed to be a gap in the research. And so what brought it back to the forefront and this sense of more urgency? I think I can guess, but.
Yeah, so it's a good question, and as you mentioned, the first publication on this that I'm aware of is in 1930 by an American scientist, actually. And at that time, it was noted, just like we noted here, that it's present in marine fish, not in freshwater fish. It was also noted that it was present whether the fish was fed or not. So we know that this is not related to digestion. And there was a pause of about 30 years before the next work was published on that. And that work was, with all respect, largely confirmatory. There was a few more papers in the 70s and 80s, a very interesting paper in 91. And then late 90s, when I picked up working on this, we started looking at, first of all, how are these high concentrations of carbonate occurring in the intestine? And second, we got to the question of why are they doing this? And we published the findings on that in 2002. And I would say late 90s, early 2000s is when the field sort of took off. I mentioned the paper in 2009 where we first estimated that this is probably very important globally. Sort of kickstarted some interest, but I want to say that it's not actually until maybe the last 4 years that this is really gaining momentum internationally. And the reason for that is something that Ben alluded to. It's the interdisciplinary work that we're doing now where we're combining biology and geology, because that's necessary. I'm a physiologist. I look at how fish work. The same is true for Rachel, whereas Amanda is a geologist, and she looks at these biogeochemical cycles. So by combining those two fields, we've got an insight that we haven't had before and that we wouldn't have had otherwise.
And you talked about the difficulty in researching this and just getting fish to survive so you can look at them. do you want to talk about how you found the back belly rosefish? Because that seems to be a little bit of the star of the show.
Yeah, so in many cases, when you make these advances in science, it's not always something that you sit down and think about and plan carefully and then execute. Sometimes things happen by chance, and you gotta capitalize on whatever observation you have and jump on it. And this is an example of that. It's probably no secret to many in here that what I do in my spare time is fish. So I work on fish when I work, and then I also fish when I don't work. And I was out recreationally fishing and catching black-belly rosefish in 1,300 feet of water, and I saw one of them shake off the hook both side. And normally when fish are brought up from that depth, they're kind of done. But this black-belly rosefish took one look at me, or at least that's how I saw it, and then it turned its head down and started swimming. And it just kept going. And that made me realize that these fish are competent and healthy when we get them to the surface. So I went back, and then the first thing I did was, of course, ask for a permit to go collect these for scientific purposes. Took a cooler out the next time, realizing that temperature was really, really important to keep them alive. Tried with a single fish, got it into Crandon Park Marina right across the street here, where it was fine. I then filleted and had it for dinner that night. But at that point, it was clear that we had a good shot at working on these fish in captivity, and that's what we've done since.
And so that was an accidental discovery. But what other kind of research or advances in technology have happened that have allowed you, again, to do research at this very deep level and hard to reach, and you can't just like jump down in the water and snorkel down and find what you're looking for?
Yeah, so that's a good question, right? So obviously in this case, we got to angle them at depth, very strong currents. It's not as easy as it looks, and you got to combine that with, what we have on this facility here, this campus here, in terms of ability to look at the physiological processes, the biochemical processes. So you got to combine all these things to make advances here. We've looked at pretty much all aspects of this from a fish biology perspective, and we're now getting into what it means for the carbon cycle. So we know a lot about how they do this at the molecular level, the cellular level, what the implications are for organ function and the intact animal, and how it varies with environmental conditions. So different temperatures, different salinities, different CO2 levels or pH levels. You're going to hear more about that shortly. So I wouldn't say that there's a specific technological advance or advantage that has allowed us to progress here. It's more a combination of the two fields that we talked about.
Okay. So then the carbon cycling. This to me gets complicated. Amanda, if you could explain that, so now you've got these fish, and I think the first time, and maybe this is the joke that Ben was going to avoid, pooping in the ocean and you guys collecting this stuff. And I know don't call it fish poop, but then why does that carbon cycle carbon? How does that work?
Yeah, so we call it ichthyocarbonate. Ichthyo for fish, and carbonate for the mineral they produce in their intestines, so it sounds very professional, ichthyocarbonate. I'm going to go ahead and share some slides, because when we start thinking about how marine fishes influence the global carbon cycle, you look at the state-of-the-art knowledge, and you quickly realize there are 3 knowledge gaps that really prevent us from quantitatively telling you how marine fishes impact carbon cycling. And so these 3 knowledge gaps, especially the first 2, are the ones that we've been tackling together in this interdisciplinary collaboration. And so ichthyocarbonate is very important in terms of how marine fish influence the global carbon cycle, but it happens through multiple ways. And so the first thing we need to do is understand how much they produce. Martin's already touched on that. But also what it's made of and where it ends up. Does it end up in the sediments in the shallow ocean or the deep ocean, or does it dissolve in the water column? And so to build understanding on these knowledge gaps, we've been working together to try and determine these factors. And so I'm going to start by talking about each one of those knowledge gaps, if that's okay, Jenny. The second one will hit your question on carbon cycling.
Okay.
Okay, so Martin did a great job of explaining how we've had a lot of technological advances since 2009 that showed us the global fish biomass, especially at mesopelagic depths between like 200 to 1,000 meters, it's increased, our estimates have increased by a factor of 2.7 to 9.5. So that means that initial estimate from 2009, where we thought there were about 2 gigatons of fish in the sea, could be as high as 20 gigatons of fish in the sea. And many of them look like this photo I'm showing you here on the lower left. They're very small. They have really interesting bioluminescence because they live at deep depths where there's no light. And so when we realized that we needed to revise those global estimates, we just did some algebra. So I'm going to show you a table and walk through it. On the left,
This is for me, right? You're going slowly.
in the blue, you'll see the increased factors of global fish biomass. So the 1x, that's referring to the 2009 estimate. That means no change. There have been a few studies, one study that suggested a slightly smaller biomass, but the majority suggested a much larger one.
Sorry.
And so each one of these rows in the table is a case study. It's a possible outcome of the changes in the global fish biomass. And so using that larger global fish biomass, we just multiplied the rate of production by this larger amount of fish in the sea. And so on the table here, you can see a low estimate and a high estimate for production rates because there's a little bit of uncertainty in the daily production. But if we're conservative, we can see that this larger fish biomass is responsible for 0.17 to 0.47 petagrams of calcium carbonate carbon. Now that's a really strange unit, so I checked how much a blue whale weighs, and this would equate to something like 500,000 to 1 million blue whales, which are the largest whales in the sea, so it's kind of a lot. And if we compare that, oops, wrong direction, to the carbonate producers that we think of, the typical carbonate producers in the ocean, things like corals or microscopic plants and animals like coccolithophores and foraminifera, they're broadly thought to be the number 1 and 2 producers of carbonate in the sea. And we can see that they're producing 0.1 to 0.14. So fish are producing as much, if not more, than the top carbonate producers in the ocean. So this is a really big change in our understanding of how much carbonate is produced in the oceans. And much of it is attributable to mesopelagic fish. To get to your question of how does this influencing carbon fluxes, I made a diagram that I'm going to walk through, and there's a few equations, but I promise I'll explain all of them. So this is the ocean, and the main thing we want to understand when it comes to composition is where is the carbon coming from? So carbon in the ocean is present as DIC, dissolved inorganic carbon. This can be CO2, bicarbonate, or carbonate ions. And this is broadly the characteristic that allows the oceans to buffer CO2 that's in the atmosphere. And so there's 2 processes that influence atmospheric concentrations of CO2 on submillennial timescales. There's the biological pump and there's the carbonate pump. And I want to walk through both of those and then explain how marine fish interact with the 2 different processes. And so first I'm going to start with the biological pump. And when you think of the biological pump, you should think of the food web. And at the base of the food web are photosynthetic plants. And these plants, this is the equation I promised I would explain. This is CO2 and water plus light is how these plants get their energy and make their organic biomass. And so that CH2O is the organic matter. And then they release oxygen. And over time, the cells, so we're removing CO2 from our seawater DIC. Those cells actually sink through the water column, and that's a process that transfers carbon from the surface ocean as organic matter to the deep ocean. But they also can be eaten, so consumption by zooplankton and secondary consumers like fish can repackage that organic matter into things like fecal pellets, and broadly speaking, 1 to 10% of what's produced in the surface ocean makes it to the deep. The rest of it kind of gets recycled through this remineralization term. So overall, when you think of the biological pump, it's removing CO2 from the surface ocean and therefore drawing down atmospheric CO2. On the flip side, you have the carbonate pump. This is where carbonates are produced. So the formation of carbonate minerals, calcium carbonate minerals, also influences atmospheric concentrations of CO2, but in kind of an unexpected way. So you can see that calcium and bicarbonate are removed from seawater to produce the mineral calcium carbonate, but it actually releases CO2. So this is a counterbalance to the biological pump. So this is an example of a microscopic plant called a coccolithophore, and the popcorn-shaped one next to it is a microscopic animal called a foraminifera. Those are the top two in the traditional perspective, producers of carbonate in the sea. And so they also remove carbon from the seawater DIC. They sink through the water column, and that is exporting carbon in a different form, in an inorganic form, to the deep ocean. This may dissolve, end up in the sediments, but the net effect is that there is some CO2 released back to the surface ocean through this precipitation reaction. And there's great work in the 90s by folks who showed that for every mole of carbonate produced, about 0.6 moles of CO2 is released. So it's not exactly a balanced equation with the biological pump, but they both influence CO2 transport. And marine fish actually interact with both of these pumps. So they're eating food and drinking seawater, just like Martin said, to stay hydrated, and they're producing the ichthyocarbonate and fecal pellets, which then sink through the ocean and contribute to both of these fluxes. And so one of the key things that's really special about fish is they actually use a lot of carbon to build that carbonate mineral from the biological pump, from their diet. The vast majority of carbonate producers use less than 1% or less than 10% of dietary carbon to build their shells and skeletons, and marine fishes use anywhere from 90 to 95%. And this is one of the big questions why composition matters and why fish impact the carbon cycle, is we need to understand in which direction the CO2 fluxes are driven by the formation. Did that get to your question?
It did.
Awesome.
But I've got, when you're done, follow-ups.
Cool. So after we've produced it and we've learned a little bit about its composition, and a lot of this has some really great work done by the students in our group doing geochemical analyses and physiological analyses, we then have to ask ourselves, with this is the reliance on dietary carbon. We use the mass spectrometers in the lab to measure this as like a fingerprint, which is very fun. But then we have to ask ourselves, where does it end up? And so on the right, you can see I have a little schematic here of a fish in the open ocean. That sinking ichthyocarbonate can dissolve. It can dissolve at many different depths. That's going to be an important control on where the CO2 and the carbon ends up. But it also can end up in sediments. And as a geologist, it's very exciting because marine fish evolved between 300 and 400 million years ago with this capacity to produce ichthyocarbonate. So what I really want to know is, can we find it in the sediments all the way back, when dinosaurs were on the planet? I think it would be a really cool project. And so I just wanted to take a quick second to show you what ichthyocarbonate looks like in the sediments at two different scales. So on the top, these are scanning electron microscope images. It's a microscope that looks at things at a micron scale. They're very, very small, and they have unique shapes, which help us diagnose that this is carbonate from a fish. And then the most exciting one was a recent paper from this year from Woods Hole, where they showed ichthyocarbonate in sediment traps, which are essentially specialized buckets that collect what's sinking through the water column. And they found ichthyocarbonate at 500 meters, but also 3,200 meters. And so this means that fish are influencing carbon fluxes throughout the water depths in the ocean. And so those are the three main knowledge gaps that we're working really hard to address together so that we can give you a quantification of the direction, magnitude, and timing. But that's kind of a good summary.
So those knowledge gaps, like I think one of my questions for you was, well, you can't study the whole ocean, so how do you get at this? We talked a little bit about citizen scientists. Maybe you can recruit anglers to catch fish in different locations, but even that's just a drop in the bucket. So how do you fill these gaps?
Yes. So as Martin mentioned, there's 13,000 species of marine fish that osmoregulate, making ichthyocarbonate. So it would be really tricky to go through and try and do controlled experiments on every single one. And so what we've done as a first pass is try to understand how environmental conditions like temperature, fish size, and fish lifestyle impact the production rate, composition, and fate of ichthyocarbonate. And so we try to select fish species that are representative of an active lifestyle or a more benthic sort of, I don't want to say lazy, sedentary from my biology colleagues, sedentary lifestyle. And then also look at things like fish size, which, fish size changes throughout the lifespan and also with different species, so.
And then we talked a little bit about this earlier too. I'm sorry, I keep referring back to the conversation we had leading up to this about what you know and what you don't know, and how that is sort of either complicating the research or helping the research?
So what we know and what we don't know. I think we're making progress. So we've learned a lot about production rates through our controlled experiments, a lot about composition through the analyses we're doing with our students in the lab, and through FATE. But I think we still have questions about the deep water fishes, as Martin pointed out, about the actual mesopelagics that do the diurnal vertical migration. And also I'm really curious about the sedimentary record question. I think we need to understand how much makes it to the sediments and what form, and then what happens to it once it gets in those sediments, because the environmental conditions in the sediments are really different than the ocean. So maybe they change in ways we don't expect.
How would you figure out how much makes it down to the very bottom of the ocean?
So first you have to understand how much is produced. Then you need to understand its composition and how composition impacts dissolution rate. So Martin had a postdoc in his lab, Eric, who did a really nice study looking at size, specific gravity, which is like the density of the mineral, sinking rate, and then measured the dissolution rate, I don't know how many, hundreds. He worked really hard on that study, of dissolution rate measurement so that we could create a curve, so we could understand how carbonates of different compositions and different sizes, how quickly they dissolve, and then they designed a really nice model to predict where they end up. And so that was our first pass at estimating the fate.
Okay,
Yeah.
okay. And I was in your lab for some extraction of the ichthyocarbonate, but you do a lot of other stuff in there. And one of the things that I have talked about on other panels is I'm always amazed at the things you create to do your science. And there were a lot of things in the lab. I mean, just discovering the right fish is a big thing, figuring out how to keep it alive. But what are some of the things that you had to teach yourself to get to this point, if that makes sense? Or the things you had to build?
I won't say teach myself, because I have really generous colleagues who are teaching me all about fish physiology, but I think that Dean Kerman really described it well, that we had to build an interdisciplinary language and make sure we were talking about the same thing. And I think maintaining a level of curiosity, because at our weekly lab group meetings, I'm constantly learning something new about marine fishes. But we also do a lot of designing new components. So I'll let Martin and Rachel talk about the tank buildup. That our students have a lot of plumbing experience now. But one of our outreach events is we teach a 3D printing and design course, and we've found it really useful because we need a special bracket to hold a valve in exactly the right position. We can design it and 3D print it and make life easier for that thing we have to do 10 times a day. So that's a good example, I think, of learning something new and making life and research easier.
Got it.
I don't know if anyone wants to chime in about plumbing.
I mean, we could maybe add that the work we've done on FATE involves developing methods to detect the specific gravity of these compounds. Also at remote locations, we have sophisticated equipment that can do it here, but when we travel to, say, Panama very remotely, we're not able to do that. So we had to develop techniques for that. The same for the dissolution rate measurements. We've developed a couple of different techniques that are complementary and gives us, I think, realistic rates for dissolution. But it's important to note that those methods are developed by us to address these questions. So there's a lot of that. But I agree with Amanda completely that the common language really was probably the most important thing.
Yeah. So, Rachel, now how do you take this and put it into a climate model? How do you use all what these findings to tell you how it's going to impact climate change or impacts from climate change?
So that's a very good question. I'm going to go through that in my slides. But yes, so Amanda pointed out a number of knowledge gaps, and I'm just going to add another layer of complexity on top of that and throw climate change into the mix. So I'll switch over to my slides here. There we go. So I know that to this audience, climate change is very well known and well studied. We are expecting more than a doubling of CO2 in the oceans by the end of the century, and that's going to co-occur with temperature increases of up to 5 degrees Celsius. So these are very drastic and rapid rates of change. This is illustrated in these figures here on the upper left. Here we see CO2 kind of in a more near future setting, and then by the end of the century, the darker colors being higher levels of CO2. And in the bottom left, we have temperature projections out to year 2100 under a different CO2 emission scenarios, least conservative to most conservative. So the top is probably closest to where we're at now. So we're expecting these big drastic changes. As we know, they're going to have impacts on ecosystems. So one of the questions is, why does this matter for ichthyocarbonates? What is the link? And what we really have to think about is that when we have changes in water chemistry and temperature in the water, that's going to impact some of the physiological drivers that Martin mentioned that form these carbonates in the intestine. So that's one aspect that we have to think about. And the way we can get at this question as experimental biologists is to run lab simulations. So we can take fish from current day and then put them in water that mimics these future conditions, and then look at things like production rates of carbonates. And our group, as well as a couple of groups worldwide, had done this. And the overall patterns that we're seeing so far is that both temperature and CO2 in isolation lead to an increase in ichthyocarbonate production. So the fish are making more when they're exposed to these stressors. So that's something that we have to think about that, again, adds that extra layer of complexity. We know this from looking at both individual fish, taking directly from the intestine, and from experiments where we look at collection from the tank bottoms of groups of fish, so both scenarios. And so in addition to increases in production, so there's two main knowledge gaps in this area of research to build on Amanda's gaps. First is that we only have this information for a very small group of species. There's only probably 3 or 4 groups in the world that's currently done any sort of experiment on this, so we have very limited species representation, which is a little bit of a problem because there's a lot of species variation across fish in the way these carbonates are produced and the composition of them. And then I would say the second knowledge gap is so far, again, this field is a little bit newer, we've been doing a lot of isolated stressor studies. So we'll just expose fish to either high temperature or high CO2. But to my knowledge, we've done a pilot study the last 2 years. I think we're one of the only groups that has combined these stressors together to look at how they're going to interact. And this is really important because none of this is happening in a vacuum in the future. It's happening together. And sometimes when you combine stressors together, you can't always predict how the outcome is going to go. You might think 1 + 1 is going to equal 2. It might be that 1 + 1 = 4. You never know how these things are going to interact. So that's one major factor that we have to think about going forward. And then building on that, so we're now thinking here about one fish, a group of fish. What I'd like to do now is expand a little bit and think more about communities or populations of fish and communities. So now Amanda touched on different lifestyles. So we have fish that are pelagic, we have mesopelagic fish like the black belly rosy that you see here. We have benthic fish, or I guess we can't say lazy, sedentary fish. And so like Amanda said, primarily we think of the ichthyocarbonates going in kind of 2 directions here. After they're excreted from the fish, we're thinking about where do they actually go. One is into the sediments, as Amanda mentioned, so that's one carbon storage area. But then we see dissolution. So this is what Amanda was referring to. We have our calcium carbonate, and as it dissolves, it goes back into the ionic form, releasing both those calcium ions and the carbonate into the water column. And Amanda mentioned buffering, and part of what this process does is add that carbonate back to the water and buffer. So, again, we get back to this question of what happens in future oceans. And so that's something that we've also looked at in the pilot study that we did, thinking about dissolution rates in future oceans. So we've designed a study that was similar to what I've already described in the prior slide, but instead of looking at production, we're taking carbonates from toadfish and putting the carbonates in different oceanic conditions that mimic future oceans. And then we're measuring dissolution rates. So just to give you a little bit of information on this figure here, we have dissolution rates on the y-axis. We have control, or what would be considered current day, a high CO2 treatment, a high temperature, and then a combined treatment. And I hope what you can see here is that any of these treatments that involve elevated CO2, or what would be representative of ocean acidification, are leading to a more than 4 to 5-fold increase in the dissolution rates of the carbonates in those treatments. So this suggests that in future oceans, the carbonates are likely going to be dissolving at a faster rate. And if we think about what Amanda mentioned in terms of sedimentation versus dissolution, what this is likely going to mean is that there's less of the carbonates going into the sediments and more dissolving in the water column. And this might be interesting in the terms of a community-based thinking, because if you have an area where there's a lot of fish or high fish biomass, the excretions from these fish may be able to buffer some of the local changes in pH that are occurring as a result of predicted climate change.
So I have to say, I'm a little humbled by all this because I thought I understood it, and now I'm realizing that there's still a lot I don't. But so when I was asking about like climate modeling, I think I was thinking like, oh, you understand this now, and how does it change your model and your forecast? But you're still you're still putting it into the model. I mean, I'm not sure I'm asking the question correctly, but you're not at the point yet where you're issuing forecasts. You're looking at how climate change is going to change the fish, not how the fish could change the ocean.
Well, that's certainly, we want to get to that point. And I think what we're doing now is kind of getting the building blocks to be able to do that. Because in order to build a model, you have to have inputs that can go into it. And that's what we're working on now. So quantifying production rates under these future scenarios, quantifying dissolution rates. Once we have that information, then we can reach out to more of our interdisciplinary teams, biogeochemical modelers, and we'll talk about fish movement patterns in a moment with climate change. But then we can start kind of thinking a little bit more about how we can integrate these factors into models. I don't know if Amanda wants to build on that. She's
Sure.
She's the carbonate modeler more.
The modelers want an equation that says when temperature changes in seawater from X to X, dissolution rates increase this much. And I think we're still learning about those rates and their responses to different environmental conditions. And so a big push is to try and get those equations locked down, because right now fish are not in fully coupled or system models. So when people are modeling climate, there is no contribution from fish because they're missing a lot of this information about how much is made and where it ends up. And so we hope we're going to fill that knowledge gap and give them the quantification they need to parameterize fish in the models.
So like them, I like quantification. Do you have any idea, since this huge thing is missing from the models, like how much influence it will have when you figure these things out?
That's a great question, Jenny.
That's a good question.
I think, do you want to us to? Many of the things work in opposing directions, right? So you produce more with high CO2 and high temperature, but it dissolves faster. And so we really need to integrate across the time scales and the perspectives in order to get that, what direction and how much are they influencing carbon's cycling in the oceans. So we hope so.
Yeah.
Martin, did you want to add something?
Yeah, I mean, even considering current day oceans, what we've shown is that this is probably the biggest producer of carbonate in the global ocean, and they're not considered in these fully coupled models. So with all due respect to the people that are working on these models, I think they're probably not 100% accurate because they are missing this large component.
Okay, that I get.
So we don't even have kind of the present day fully locked down in terms of their contribution. So it makes it even harder to project it out into the future. So we're kind of chipping away what we can now, and hopefully, in the next decade, I hope that this gets into more of these models, and then we're able to gain resolution.
So, and one of the questions I had for you, because I've written about that, is climate change is already affecting the movement of fish and where they are and where they spawn and all those things. Martin and I did a story together. And so you talked about how different ocean conditions could affect how the fish produce the ichthyocarbonate. What happens when the fish are moving? Is it sort of the same thing, or?
It's a great question, and that's my next slide, so I'll transition over. So I've now shared with you that, excuse me, I've now shared with you that we know that these conditions increase production and that they also increase dissolution. So once they're excreted from the fish, they dissolve faster. So now, again, we're going to add another layer of complexity. We know that climate change has a very broad expected negative impacts on fisheries. So, for example, movement patterns are expected to change as a result of climate change. I have an example of this in the upper left diagram, just showing that in general, species are expected to move poleward, and that's because in the equatorial regions, it's going to get warmer, warmer more quickly, and the species that can move are likely going to kind of creep upwards, up and down away from the equator. So the top panel here is species invasions per half degree of latitude by year 2050, for example. This is just a projection. So in general, poleward movement away from the equator. A second component, if we just take a look at an individual species, so I've pulled this map from Aquamaps, which is basically a program that Gabriel Regenduve in our school, Rosenstiel, I guess he contributes to it or he runs it. He's doing a Sea Secrets on April 7th. So if you're interested in this, please stop by there. But he produced this map, and this is Atlantic bluefin tuna. In the left, we're seeing the probability of finding them in that area now, and then in 2050 is the one on the right. So we are expecting changes to things like migration patterns and movement patterns of fish that can do that. So that's something else to consider. So fish are not going to be in the same places that they were before. The second major factor to consider is that we are expecting on a broad scale biodiversity declines, and that can kind of get bent into two separate categories. One is thinking about species richness, so that's the different types of species that are there, that's expected to decline. And then species abundance is the number of those types of species, so how many flounder, how many toadfish, that's also expected to decline. So we have these two factors also that we have to think about that are going to be changing. So this again is from Gabriel in the top, just showing changes in species richness expected by 2100. So we can see there's a lot of red there, so we're expecting a lot of loss of species richness. And if we're thinking about a simple example of a fisher person catching animals here, we can kind of see this conceptually, where we have not only a loss of the different types of species, but the total number of species as well. And then the final thing I'll mention also relates to what Amanda talked about earlier regarding fish sizes. So by and large, we are expecting in future oceans for fish to generally be of a smaller size. There's exceptions to that rule, as always in biology. But overall, the size of fish is expected to be a little bit smaller. And why that matters for ichthyocarbonates is some work done by Eric Folkerts from our group that you can see here in the bottom right, where on the bottom we have fish size, so increasing fish size. We looked at 3 species here at Rosenstiel. And what Eric wanted to know was, do bigger fish make bigger carbonates? And the answer was yes. So in future oceans, if we have smaller fish, that means smaller carbonates, which can affect the way that those carbonates move through the water column, might impact sinking rate and then thus dissolution rate, and whether they end up either again in the sediments or dissolving. So those are a lot of the factors, those are just a handful of factors we have to consider on top of everything else when we're thinking about the future.
And then I think this question leads in from that. In terms of fisheries management and protections, how would understanding this impact those? Do you want to protect fish who are in a certain area because they're important to the cycling, or what?
I think that's a bit of a tough question, but I would argue that if we could use the idea that they could provide alkalinity in local environments by their excretions and the fact they do dissolve more quickly, it might buffer changes in areas that are particularly fragile. So maybe adding an argument that if something's already protected as an MPA, maybe the area surrounding it with fish, it'd be useful to make sure certain fish are there of high biomass to kind of buffer those changes in alkalinity in the water column. So there might be areas with high coral cover that would benefit, for example, from having an increased alkalinity. So it might help other species in ecosystems. So that would probably be the main argument I would make.
Okay. So then you all talked a little bit about the interdisciplinary work. I wanted you to kind of go back through and explain a little more specifically, like how your work interacts, and also how each of you got involved in this. I mean, Martin loves to fish. We've talked before. I knew you grew up fishing, but this to me is like for me, a niche science. It's not for you. It's like the main science. But how did you get here?
What, into science in general, or this particular question?
This kind of
Okay.
science, yeah.
So late 90s, I was doing a study on something completely unrelated, and I was measuring the composition of intestinal fluids, and reported 70, 80 millimolar of bicarbonate, which is very, very unusual. There are very, very few biological fluids that have that composition. And so I started asking the question, what is up with these high bicarbonate concentrations? And found the paper that we discussed earlier from 1930 and a few others from the past century. And there was a transport model proposed. When I say transport model, I'm talking about the cellular mechanisms that allow for these high bicarbonate concentrations. There was that published, the only one at the time, and it did not, thermodynamically, it did not make sense. So then I started asking the question, where is this bicarbonate coming from? How does it get there? And so that was my introduction into this field. This was all worked on in Canada, actually, on Vancouver Island at a place called Bamfield Marine Station.
So you had that question. Did you realize like the big consequences?
No, not at all. No, and I think that goes for a lot of scientists and a lot of scientific questions at their emergence. You're curious about something, you start digging into it, and at that point you very well may not realize where that's going to go. And I think that's just a general plug for supporting basic science that not necessarily is focused on solving a current problem, but can expand our knowledge and thereby position us better to address future problems. And that's a very general thing, but.
And I know that we first talked about this, I think, 3 years ago. So it also, hard science takes time.
That's, we first published this, as I mentioned earlier, 2009, that this is a big deal. This needs to be considered for coupled modeling. And it has, to be perfectly honest, been very frustrating to see how slow that has been adopted by people that really should be adopting it. And it's not, as I mentioned earlier, until the last, I would say, 3 or 4 years when we started working together, that we're getting some traction on that. And I think that, again, points to the importance of interdisciplinary work that Ben alluded to. If we're not speaking the same language, if I'm talking about this to my fish physiology colleagues and my fish biology colleagues, but the biogeochemical modelers are not getting it, then we're not advancing the field. So that is essential for this progress.
So how did you and Amanda find each other?
So Amanda should answer that.
When I first started, I got a new mass spectrometer that could measure concentrations of elements, and someone had graciously referred Martin to me to measure magnesium content in something. It actually escapes me what it was. But he just sort of offhanded mentioned that marine fish make carbonate sediments in their intestine, and my jaw dropped. I was like, I'm sorry, I'm a carbonate geologist. I didn't know fish make sediments. And that conversation sparked the collaboration. I think just the willingness of them to teach me more about fish physiology, and for me to be excited to try and use my geological tools to answer some of these questions. It was actually a chance conversation. I think Rosenstiel's a really special place for that, because we have so many different departments of people working on different questions, all right next door to each other.
And Martin mentioned speaking the same language. I mean, just now when you said sediments, I'm like, oh, okay, that would make sense to me. If he's calling it something else, ichthyocarbonate, well, maybe you would get it, but I might not. And then Rachel, you got roped into this.
Yeah, so I originally I was interested in corals when I was trying to go to grad school, and then Martin, after recruitment weekend, I got an email saying, would you consider working on fish? And I thought, I really am into the physiology and I'm into climate change. Sure, I'll give fish a shot. And so I started I did my PhD with Martin starting in 2010, and I just became really fascinated with fish and their physiology. I've always been curious and wanting to know not only the what, but the why. And physiology is really the best field of study in order to get at the why. And just the idea of trying to project what's going to happen in the future with all of these changes that are happening in the environment also was just fascinating to me. And then Amanda and I overlapped here during graduate school, so we actually knew each other before the collaboration started. And so it all felt very natural and organic for us to work together. And Amanda says she's learned a lot from us, but I would also say that I've learned a lot from Amanda. I think she's probably explained stable isotope ratios to me probably at least 10 times at this point. So there is this great kind of two-way information flow. And, to just briefly tell a story, we went to Panama to study something completely different. And just to see everybody in a kind of a natural environment get really excited, not only about what we're doing, but Amanda, was it pillow salts? We were out doing a hike, and Amanda sees this geologic formation, and she just lights up and it's just so excited. And, I shared that excitement. I didn't know what it was. To me, it was a rock, but when she explained it, it was really cool, and I've just gotten this whole new appreciation for geology, and yeah, I would say it goes both ways.
Excellent. So we hands are already raised, so I think we'll move on to the question and answer.
Well, thank you very much. That was really interesting. Thank you. Here come our students, Sarah and McKenna. Each one is going to take a aisle and raise your hands, and they'll get to you. When you have asked your question, please return the mic to them so they can keep moving. All right, right here. Start with.
Yeah, fantastic presentation. Really exciting work. There are some very high fidelity records in anoxic areas like the Cariaco Trench that Peterson worked in for so many years. I wonder whether you can see entire ice age cycles in the sediments that he has collected there, and would that be helpful to you in understanding what the flux is through the whole cycle? You may not get as far as the climate change is going to take us, but maybe you can see some indication.
That's a really good question, and I hope NSF funds my proposal that's currently in review. I really want to know how can we identify carbonates produced by fish in the sediments, because then we can start answering questions like that and looking for places where we have high productivity and high preservation. One of the things I sort of suspect is once it gets into the sediments, it might not look the same. And so we're hoping we get some funding where we can quantify how does it change as it reaches different sedimentary environments to sort of set up like a Rosetta Stone of this is definitely ichthyocarbonate, and now we can understand how it changed through time. It's a great idea.
Okay, I'm not a scientist, but I do work at Mass Academy. And I'm wondering about the role, if you know that sharks that go up and down the water column, that dive to great depths and then come up to the surface, play in this, and whether cetaceans have a role as well.
So sharks and cetaceans, so neither of them are producing these carbonates. It is specific to the bony fishes, the teleost fish. Yeah. It is a very interesting question though, because we have played with, we've done experiments on some of the shark species. And what's really important to recognize is that they can produce these carbonates, but only if they are dehydrated. So normally sharks and skates and rays do not drink seawater. But if you dehydrate them experimentally, they will drink seawater, and when they do, they produce these carbonates. And it's important for two reasons. One, it confirms that this is in order to stay hydrated. That's the reason fish are doing this. And two, it shows us that ancestral vertebrates, sharks are a representation of those, were able to do this. So we can go back 3-4 million years or more and say fish at that time were producing these carbonates. So it has very, very important implications for the predictions of whether we're going to see these carbonates in historical sedimentary records or not. Lamprey, some of you may be familiar with lamprey, current living organisms that are again representing ancestral vertebrates are also capable of doing this. So even though sharks do not do this on a normal basis, your question is interesting because it allows me to share that we know that this occurred a very, very long time ago from an evolutionary perspective.
Yes, I'm curious if the ichthyocarbonates, is there any connection with DNA? Like, can you track DNA from the different fish producing it?
So we've done a little work now on trying to identify the microbes that are associated with these ichthyocarbonates. So there actually is a rich microbial community associated with them, and it's unique. We know it's different from seawater. It's different from what's found in the intestinal epithelium of these animals. And yes, we've identified those by looking at the DNA sequencing, the DNA sequences of that material. In addition to that, there are about 100 proteins that are produced by fish that control the formation of these carbonates. It's a complex scenario, but this is so important to fish that it's not just a matter of inorganic chemistry. They control it biologically by these proteins. And so certainly the proteins can be isolated from these carbonates, and that it's a great question, it's a good idea. That may be a source of identifying or labeling these carbonates. The question, of course, is how stable are those proteins going to be, and how stable is that DNA?
Thank you. I enjoyed the presentation. I'd like to know whether fish evolved in freshwater and evolved this as an adaptation to moving to salt, or might have been in the other direction. I'm also wondering what they do with all the sodium that they drink.
Okay, so that's a two-part question. The last one, I'm gonna save the last one because that's the easy one. The first one, the current view is that fish originally evolved in seawater. Some invaded freshwater. There was then a mass extinction of the seawater species at the time, and marine environments were then re-invaded by these freshwater colonizers. And it's probably for that reason that all marine species are capable of this feat and drinking seawater, because that is the true ancestral state of marine fish today. It's probably also why we see that capability present in the sharks, rays, and skates that we talked about, and also in lamprey. To your second question, the sodium that's coming with seawater, and it's a very good question because, of course, sodium and chloride are the two most concentrated ions in seawater. And they're taken up by the esophagus before the water reaches the intestine, and then are immediately excreted by the gills. So the gills act as a salt gland. The salt glands you see in birds or turtles, the gill play that role in marine fish.
Hello, guys. Thank you. I'm curious, is ichthyocarbonate production tied to metabolic rate? And if so, is there any concern with using these more sedentary benthic species for our estimates? And do you have any thoughts on resolving that?
Thanks, Jonathan. That's a fantastic question. I don't know if you've been talking with some of the other grad students in the department, but yes, there's a very tight link. So drinking to rehydrate the animal is a function of metabolic rate. So the greater the metabolic rate, the more these animals have to ventilate. The more they ventilate, the more water they lose. And as a consequence, the more water they must drink to replenish that loss. So when we're working with benthic fishes, the rates of ichthyocarbonate production we're getting are lower than you would expect from more active, say, pelagic fishes. We are right now building a relationship between metabolic rates and ichthyocarbonate production. And that relationship is actually, it's going to help us a lot refining the global production rates of ichthyocarbonate, because metabolic rates are so much easier to measure than the rate of ichthyocarbonate formation. And in fact, there's a wealth of literature reporting metabolic rates for a very wide range of species that we can then use to sort of refine our estimates of global production. So it's a very, very good question. Thank you.
I was going to say, can I jump in on and add on to that in terms of thinking about environmental factors like temperature. Metabolic rate actually scales with temperature. So as you increase temperature, a species' metabolic rate is going to go up pretty drastically. So that's another aspect of this that is interesting from the climate side of things.
This was fantastic, by the way, so thank you. So I have a budding marine scientist with me here today, and what I kept thinking about was that you're talking about the future oceans that she'll be studying. So I have one comment and then one question. I hope you'll consider doing more outreach and engagement with the future scientists of Broward and Miami-Dade, because I know that they're excited to get involved in a lot of this. Wonderful. So my question, I'm actually a communications and engagement professional, so I'm curious about this interdisciplinary language that you talked about, and what was that like to create or come across, and the experience once you did create this common language?
I would say it's still in progress. We're all reading papers from the different fields of study with each other and discussing them. So the geology students are presenting papers on physiology. The physiology students are thinking about stable isotope ratios. And I think through discussing the papers together and asking questions and having a culture of curiosity is really helping us to build that common language. But even on Monday, we talked about something that blew my mind. So I think this is in progress and continual development. I don't know if anyone wants to add anything too.
I think for me, it's put in perspective. When you do a PhD and beyond and graduate, you start kind of getting narrower and narrower in your focus. And what working across disciplines has really made me think about how I'm presenting my own science to make it more accessible. So it's not only just within scientists, but at events like this, thinking of like, how am I going to explain this in a way that's going to make sense to more than just my immediate colleague group. So for me, it's been very reflective in that respect.
I'll say that's helpful to me too.
Yeah, I'd like to add one additional perspective, if I could. I'm an economist, and if your projections are correct about the marine life moving away from the equator towards the poles, this will be devastating to the global south, primarily in Latin America and Africa, for those coastal countries. This, in turn, and I say this with some hesitation, will lead to more migration of those devastated communities north. And the precaution is, I don't want to see one of the University of Miami boats blasted off the coast of Panama.
I'm not sure how to respond to that, but I think we're all concerned about climate change in a variety of ways. It's a very serious issue, and a lot of, it's not just the fish that are going to be impacted, it's going to be the people too. And I think that's, for me, devastating when I think about the long-term impacts. So this is one small portion of that bigger picture, but it's certainly something, there's a human component to this as well.
Yeah. Hi, Amanda, I have a question for you. I'm over here. Knowing that you're studying carbonates, is there anything specific about stable isotopes of these carbonates, the carbon-13 or the oxygen-018, that's telling you at what temperature they're forming or where in the water column? What might that let you know?
That's a great question. Andrew's a geochemist, if you can't tell by his question. So the stable isotope ratios are one of the key ways that my lab group was using to trace the building blocks of carbon that are used to construct the ichthyocarbonate. And so we actually see the delta 13C values are really, really low, and they reflect the dietary carbon source. So normally carbonates, this fingerprint of their chemistry, it'll be like 0 to +1 in the marine environment. And the ichthyocarbonate can be -4, -5, -7, which as a stable isotope geochemist, you're like, what? Like, this is shocking. And the explanation actually comes from the physiology. In terms of oxygen isotope ratios, people are starting to think about that from a temperature perspective. I'm still thinking about the kinetic reactions. So there's a lot of hydration, hydroxylation reactions that are going to be kinetic fractionation. So I think there's more to be done in that respect. But it's great because we have the tools to do it here.
Hi, two-part question. The photic zone, large biomass fishes like sardines, menhaden, et cetera. Anyone looking at that? And then secondly, this is such important work, it would be great if it could be interpreted in public aquaria. There happens to be one up the road a little bit. Helicolenus, blackbelly rosefish, displays extremely well. It would be wonderful if were to get this work on living exhibit.
I think we'd love to do that. I think that would be so cool to take ichthyocarbonate to people visiting aquarium. I think about that a lot actually when I take my kids to aquarium. I'm like, oh, that one has a really good tank bottom. We could probably get samples from here. But to answer your second part of your question, I think that would be awesome.
Yeah, so the first part, we actually get that suggestion a lot for obvious reasons. But again, in terms of global biomass, it's really the mesopelagics that we should be focusing on. But that's not to say that the anchovies and similar species do not deserve attention. I completely agree. One of the, and you probably know this, but one of the issues with working on those species is that they're hard to keep in captivity. Whereas the toadfish that you all saw earlier today, they're very easy, right? So the only thing I'll say is that we've looked at, and when I say we, it's the sort of the global community that works on this particular question. We've looked at about 160 species now, and they all do this in a way that seems to be very predictable based on size, temperature, and their metabolic rate. So it would be nice to do those measurements, but I'm pretty confident that we have very good estimates on them.
Hi. This may be a very naive question and way premature, but as I listen to the complexity of what you explore, is there any way that sometime in the future, AI might be able to help you accelerate the speed with which you find new discoveries?
Well, I have an example of a really productive undergrad. She's very creative. She was manually measuring the size of these particles. So hundreds of particles per day, per tank. It got very onerous. And she actually wrote a code that would take a photograph and automatically measure the size of the particles. And once I saw that, I was like, how about the aspect ratio, the perimeter, the area? Can we get shape complexity out of this? And in a very short little project with our team, she developed something that's dramatically accelerated our output. So I can only imagine as we all start learning how AI tools can help our research, it's just going to continue increasing. I think that's just a tiny example.
Are there any octopus or squid that do this as well?
That's a really great question. We've not looked at squid and octopus, but based on their biology and how they control water balance, I can say no, they're not doing this. We've looked at crustaceans, so crabs as well. We've looked at a few other species of invertebrates, and they don't do it. It is unique to the marine fish. Actually, one correction, green turtles, they do it also. So we can assume that turtles in general do this.
Thank you so much. Any other questions?
One more.
Wait, two more. Go ahead.
So we were talking about large fish and the bluefin tuna, one of the largest. And historically, they had huge schools. They're down quite a bit. Can you track their sediment from previous years? And then maybe make some kind of assumption based on the total biomass that's available now versus then?
Maybe one way to think about it is this sediment trap study. So they've mounted these little buckets that collect sediments at 500 meters, 1,000, 1,500, all the way to 3,000 meters. And they've been doing these collections for decades. And so that paper from 2025 that had the green sediments, they found that there was a certain frequency, a periodicity, to when they would see the particulates that look like ichthyocarbonate occurring. And at that water depth and at that size, one thing I think would be super cool to do with their data is to look at the size of the particles, since we know from work that our team has produced how to relate the size of the particle to the size of the fish. We might start getting an idea about what fish actually excreted that particle that made it to 500 meters or 3,000 meters. But I'm gonna also encourage Martin to talk any comments he has about tuna.
So we actually have done some work on tuna. None of that is published yet, and we have plans to do more. And yes, they do it just like any other fish do. They produce these carbonates. They produce them at high rates. They have the unique chemistry that Amanda has discussed, so they're typical. But to your question about the depletion of, say, large tunas and other fish populations, that of course does affect the global production of these carbonates. But it's not as simple as saying, you know, we've reduced the global fish biomass by 40% and therefore production rates are reduced by 40%. Because at the same time, the size of the fish have changed. When a fish population is fished, not only do you reduce the biomass, but you also reduce the average size. Smaller fish produce more carbonate per mass than larger fish. So that's why it's not as simple as saying, you know, half the biomass, half the production. No, not necessarily. And of course, the size of the particles again scale with fish, and that affects the fate of these particles, the sinking rate, the dissolution rate, etc. So there's no question that the global fishing pressure have altered this system already, even before we fully realize it. And that is, you know, we talked about conservation earlier, that is something that needs to be considered as well. So larger fish will produce ichthyocarbonates at a lower rate, but they will be larger and they will have a different fate than those produced by smaller fish.
I was going to add, it's not completely related, but just having access to gut samples where we can identify features of the ichthyocarbonates that we collect from different species is useful for us. So, for example, if we've looked at tuna ichthyocarbonates, we can do the SCM imaging that Amanda mentioned to get the structure and the mineralogical signature. So even just having access to more species and gut samples, I think would be incredibly helpful. I know that there's been a lionfish derby that one of Martin's students participated in where they were able to get some samples, but that would certainly help us in our quest to gain more understanding about different types of species and what they're doing.
Great presentation. I have a question for Amanda. So I know you want to look at ichthyocarbonates like in the past and evidence of that. And I know that like the ocean seafloor is constantly recycling, basically. So would you focus on the Mediterranean Sea, which has like the oldest seafloor in the world, or?
That would be a great place to do field work. I would sign up. But that's a really great geology question, right? So we know that marine fishes evolved 300 to 400 million years ago, and they all have this same osmoregulatory strategy. And where do we go to find the best preservation? You know, an anoxic basin like Cariaco or a shallow hypersaline environment similar to the Mediterranean. Those would both be good places to start, and I'm willing to go.
Thank you very much. I think we covered all the questions. Hopefully you'll join us. I think several of us are going to head to the wet lab, so you're more than welcome to keep asking questions and join us over there. Thank you for joining us. Thank you for our third year. Hope to see you again next year, and join us on January 20th for our first Sea Secrets, where we'll be talking about the Galapagos, so you're not going to want to miss that. And you'll get more information since you've signed up. You'll be getting the advertisements for Sea Secrets. So thank you so much for joining us.